Peace and greetings. Bonjour, salutations. You know, welcome to U Vision. We about to bon appetit this knowledge right here. So, scientists built a perfectly self-replicating synthetic cell, right? Crazy. So this is over, we got the time frame over 1, 2, 3, 4 hours. A new synthetic organism called GCVI SYN3A contains 7 key genes that help it divide as normal cells do. It goes to the image credit, MIT Center. Okay, granted, Elizabeth Strachowski. Nailed it. Scientists have crafted a single cell synthetic organism that divides and multiplies just like the real thing. The advancement could someday help researchers to build minuscule computers and tiny drug producing factories, all out of synthetic cells. It's dope. So like nanobots, basically. Of course, that future likely won't be realized for many years to come. Quote, there's just so many ways in which this coming century of biology could potentially change our daily lives for the better, said senior author Elizabeth Strachowski, leader of the Cellular Engineering Group at the National Institute of Standards Technology, NIST. For example, Strachowski and her colleagues plan to engineer living sensors that can make measurements from their surrounding environments, monitoring the acidity, temperature, and oxygen levels nearby. These sensor cells could also be manufactured to produce specific products, namely medicines, and could potentially be placed inside the human body itself. Quote, one vision is that when the cell senses a disease state, then it can make that therapeutic. And when a disease state is longer there, they could stop making that therapeutic. You see why I'm a little stuttery? Then it can make that therapeutic. And when a disease state is no longer there, they could stop making that therapeutic. Okay, I get it now. Basically, like okay. I'm thinking maybe like a morphine, basically to ease the pain. Strachowski said other cells could be cultured in the lab and used to efficiently produce food and fuel products, while still others could be made to perform computational functions at a molecular scale, she added. But again, these are all visions for the future. To get there, scientists need to unpack the mysteries of the cell at a fundamental level before they can manipulate it into their synthetic organisms. In the study, Strachowski and her colleagues took a step toward that goal and published their results March 29th in the journal Cell. They began with an existing synthetic cell called JCVI SYN 3.0, which was created in 2016 and contains 473 genes. Scientific American reported, for comparison, the bacteria Escherichia, nailed it, coli, has about 4,000 genes, according to a statement. This bare bones cell was crafted from the bacterium Mycoplasma genitalium, a sexual transmitted microbe which scientists stripped of its natural DNA and replaced with their own engineering DNA in creating the JCVI SYN 3.0. The scientists wanted to learn which genes are absolutely essential for cells to survive and function normally, and which are superfluous. But while JCVI 3.0 could build proteins and replicate its DNA without issue, the minimalist cell could not divide into uniform spheres. Instead, it split haphazardly, producing daughter cells of many different shapes and sizes. Strachowski and her team set out to fix this problem by adding back genes to the stripped down cell. Guessing this is the back genes. A previous version of the minimal cell did not divide normally. This micrograph depicts how the cell would split into daughter cells of many different sizes. After After years of work, the scientists produced the JCVI S1 3A, which contains a total of 492 genes. 7 of these genes are critical for normal cell division, they discovered. Quote, a number of genes in the minimal cell did not have the known function, said co-first author James Pelletier. I think I got it right. Who at the time of the work was a graduate student at the Massachusetts Institute of Technology, MIT, Center for Bits and Atoms. Similarly, it turned out that some of these genes that the cells need to divide previously did not have a known function. He said, reintroducing these genes allowed the minimal cell to split into perfectly uniform orbs. Some of these important genes likely interact with the cell membrane based on their genetic sequences. Pelletier said, this could mean that they alter the physical properties of the membrane, making it malleable enough to divide properly, or that they generate forces within the membrane that encourage the split, he said. But for now, the team does not know what specific mechanisms the gene used to help the cell split. He noted, quote, our study was not designed to figure out the mechanisms inside the cell associated with each of these genes of unknown function. Tchaikovsky said, quote, that's going to have to be a future study. While researchers continue to probe the mysteries of the minimal cell, other synthetic biologists are working with even more simplistic systems. Synthetic biology exists on a spectrum from a soup of inanimate chemicals to the full glory of mammalian cell or a bacterial cell. Strychowski said, the future of the field could lead us to an innovative wonders like cell-sized computers, but for now, the work is largely driven by curiosity about how the basic building blocks of life come together. And what that can tell us about ourselves, she said, quote, how do we understand the most basic unit of life, the cell? There's something very compelling about that, Strychowski said. Later on, we can imagine all the things we can do with the minimal platform. I agree. I agree. This might start turning out like a little Star Wars in the near future. You pop a pill and you pretty much regenerate your if you got a gash in your skin, right? And they probably like put some lube over your skin. It'll probably heal that in about 30 minutes or less. By the time your dominoes get there, you done got shot up, stabbed up, rub some lube over it, order some dominoes. Have a good night. Oh man. So the next one I wanted to touch on was yes, Michigan must tell Johnson & Johnson vaccine recipients that it was developed using stem cells. Law passed by Republicans in control of state legislature prompts Democrats to accuse them of politicizing public health efforts. Residents of Michigan receiving the Johnson & Johnson vaccine must be told it was developed using a stem cell line originating from an aborted human fetus, according to a state law passed by Republicans. The requirement only applies to vaccinations funded by the federal coronavirus relief package, prompting accusations from Michigan Democrats that Republicans in control of the state legislature are politicizing the public health effort. I mean, just making it aware. I wish they would do it for all of them, but it's pretty much is publicized that you can just pretty much research it yourself. But I would rather it be publicized on TV, radio, all that good stuff. In remarks reported by the Detroit Free Press on Monday, Erica Geese, a Democratic state senator, told lawmakers the pandemic was never supposed to be political. It's disappointing, but not surprising, that you're putting politics into this process in order to scare people from getting this extremely safe vaccine. It's not really scaring people because, like, if people have like a better understanding, move along accordingly because they feel more secure. Like, you can explain it in a way that they understand and they can trust in it, then that's cool. Like, and then you don't really provide trust if like the vaccine, which is supposed to give you a small exposure dose to whatever the virus is, and then you gradually build up your own immune system. If you take the vaccine, shouldn't you be able to take off your mask? So it's like there are other areas we can provide a little more, what you call it, secure mindset security. Maybe if it would get scrutinized by different scientists, we could definitely have a debate about it. We can definitely go forward with it. Let's just have a conversation. If you want to dead everything, then that's not really providing confidence. That's the word I was looking for, confidence. Wow. The language requiring the notification is contained in the state bill dealing with the allocation of funds from the 1.9 trillion economic relief package signed into law by Joe Biden earlier this month. Each state legislator is responsible for allocating such funds. In Michigan, while Republicans and Democrats became embroiled in wrangles over moves that would have weakened the power of the Democratic governor, Gretchen Whitmer, which she ultimately vetoed, the informed consent provision slipped through almost unnoticed. According to the Free Press, the bill states that anyone who received the vaccine paid for through the 110 million appropriated in the relief legislation shall be provided with the information or informed if and in what manner the development of the vaccine utilized aborted fetuses, tissue, or human embryonic stem cells derivation lines. Wow. The bill does not specify how the information will be transmitted to recipients or will be responsible for doing so, or who will be responsible for doing so. The state health department has updated its vaccine information website to clarify the process that Johnson & Johnson used. The Johnson & Johnson COVID-19 vaccine has been produced by growing the virus in fetal cells during vaccine development and manufacturing using the PERC6 line, the website states. Even though fetal cells are used to grow the vaccine virus, vaccines do not contain these cells or pieces of DNA. The mRNA vaccines that those by Pfizer and Moderna did not use a fetal cell line to produce or manufacture the vaccine. According to information from the Philadelphia Children's Hospital, Johnson & Johnson used the retinal cell line PERC6, developed from fetus electively aborted in 1985 in the development of its vaccines. Wow, in 1985 is a long time. The company won emergency use approval from the US government last month. Catholic leaders and anti-abortion groups have raised objections to the use of fetal cells, a process common in vaccine development since 1960s, including for chickenpox, rubella, hepatitis A, and rabies. Earlier this month, Michigan Catholic Conference said use of Johnson & Johnson vaccines was morally problematic. I'm still saying though, these are still around. Chickenpox, and I don't know about rubella. I know hepatitis A is still around. Rabies, you still get that, but you have I know you can get rid of it. Most of these things still around. Michigan Vaccine Database was reporting on Monday that 3.9 million residents had received shots, of which fewer than 75,000 were Johnson & Johnson. Vaccine denialism has become popular among the right-wing conspiracy theorists in recent weeks, gaining traction as QAnon and other extremist groups lost hope that Donald Trump's election defeat was overturned. I mean, why you got to bring up that? It's not that. Just allow people to do their due diligence, like allow people to do their research. If they present their own information, then okay, yes, people should not instantly believe it, but allow them to present that information and then people can decipher through that information. And if the government wants to step in, they should allow their information to be scrutinized, like thoroughly look through everything. Come on, it's supposed to build confidence. That's not building confidence. Last one, because I'm trying to make it short. because they kind of like, actually, it's not too. Uh, why are flu vaccinated people more resistant to COVID-19? People who have been vaccinated against influenza contracted COVID-19 less and tend to have less severe cases. Is this because vaccinated people are more cautious or there are medical reasons? A study found that flu vaccinated people were slightly less likely to contract the vaccine, COVID-19. Does a flu vaccination protect against COVID-19? And if so, why? These are questions medical professionals are asking after a team of doctors led by Anna Conlon from the University of Michigan came to some startling conclusion. In a recent study published in the American Journal of Infection Control, the physicians had looked at a patient data from 27,201 Michigan residents who had taken a COVID-19 test before July 15, 2020. Of those, 12,997 had previously been vaccinated against the flu. The study found that the proportion of flu vaccinated people who contracted the coronavirus was slightly lower than among those who had not been vaccinated. 4% instead of 4.9%. That may not look like much at first glance, but it means that people who had a flu shot had 24% lower chance of contracting COVID 19. In addition, the flu vaccinated patients were also less likely to require hospitalization or ventilation for coronavirus infection, and their hospital stays were shorter on average. However, there were no significant differences in mortality between the two comparison groups. Does our innate immune defense have an influence? The crucial question for the experts is there a medical and a microbiological explanation for these results? This could be, for example, the innate immune defense, which is possibly activated by the flu vaccination. The immune defense functions independently of specifically learned antibiotic immunity, which primarily targets the characteristic spike protein when fighting COVID 19 and thus rendering the virus harmless. So, flu vaccination rates for people over 65 years in 2018 in percentage: South Korea 85%, UK 72%, US 62%, Portugal 60, Italy 52, Sweden 52, France 51, Germany 34, Latvia 7, Turkey 7%, 2016. In contrast, the innate immune defense, which may be stimulated by vaccination, consists of a number of different elements that react to infections in general, not specific viruses. This standing army of our immune defense includes, for example, phagocytes, phagocytes, all right. The dendritic cells, cells that ingest harmful foreign particles, but also various cytokines, proteins that play a role in immune reactions and inflammatory process. Some vaccinations are generally good for the immune defense, as can be seen in people vaccinated against measles, for example. Epidemiological studies showed years ago that vaccinated children still had a higher immunity to a variety of pathogens and non-vaccinated children, even a very long time after vaccinations took place. Or is it just correlation? It is also conceivable, however, that fewer people who had been vaccinated against the flu contracted COVID 19 simply because they were more cautious than non vaccinated people. More people from high risk groups like seniors and people with pre existing conditions typically get vaccinated against influenza than young and healthy people. In the United States, for example, many seniors and retirees had already voluntarily isolated themselves early last year while others were still out in the public. However, there are two indications against such correlation. Seniors typically show more severe courses of COVID-19, which was not the case with those who got the flu shots in Michigan study. A non-peer-reviewed pre-printed study from last year points more to an immunological explanation among Dutch hospital workers who had received the influenza vaccine ahead of the 2019-2020 flu season. COVID-19 occurred significantly less often than among those who had been vaccinated. And those were no seniors, and there were no seniors over 70 years of age in either of these groups. All of those studies were, and there were no seniors over 70 years of age in either of these groups. All of those studies were of the working age and had correspondingly higher number of contract counters. Does that mean you should get the flu vaccine? Aha. Does that mean you should get the flu shot now? The studies show that more research into the role of innate immune system is necessary, and there is still some uncertainty about the studies results. Protecting yourself against COVID-19 should not be the driving force behind getting a flu shot, but is always recommended because influenza can also be life-threatening. The best time to get a flu shot is usually in autumn, just before the new flu season. More importantly, if you really want to strengthen your immune system, you should check your entire vaccination status with your doctor and devise a comprehensive vaccination plan. I'm trying to get people to talk to their doctor more, talk to a therapist more. We all need it. I do too. Make sure you have all recommended immunizations and boosters against the most dangerous diseases, including but not limited to mumps, measles, rubella, tetanus, polio, diphtheria, peroteus, tuberculosis, smallpox, hepatitis A and B, shingles, were applicable, tick-borne, encephalitis, virus, and human papillomavirus. Wow, these names. Whoa. How do you come up with these names? But thanks for stopping by. Like, comment, subscribe. Um, About to start doing more summaries of these articles, but I feel like I need to give the whole article because I don't want to give no one side opinion. So I want to hear from one side, then actually give my opinion on it, so then you kind of hear both sides. Like, comment, subscribe. Thanks again for stopping by.